we've been interested in understanding the origins of lung cancer in never smokers. We've known for at least 2 decades there's an association between environmental pollution in the context of these PM2.5s, these 2.5 micron and below particles, and lung cancer risk. But that was only an association. There was a lack of functional mechanistic data to prove causation. So we tackled this problem in a number of ways. First of all, we looked at the incidence of EGFR mutant lung cancer across the planet in various countries, Korea, Taiwan, and through Public Health England data in England. And we showed that there's a correlation between rising PM2.5 levels and incidence of EGFR mutant lung cancer. So that substantiates what's already known, but specifically looking at EGFR mutant disease, commonly seen in never smokers. We then took 3 mouse models where we attempted to induce an EGFR or KRAS mutation in the epithelial tissue, and then 3 days later exposed mice to diesel particulates, PM2.5 particulates, 3 times a week for 3 weeks. And at the end of the experiment, roughly about week 10, we harvested the mouse lungs and we found more tumors in a dose-dependent manner of PM2.5s in mice exposed to pollutants compared to those not with saline control. So I think that tells us that there's clearly a promotion effect going on. The question is, how does this happen? And what we found through a series of experiments is that pollution exposure in mice and humans results in release of a number of cytokines, one of which is interleukin-1 beta. Of course, we were alerted to that because of the prior canakumumab CANTOS trial, which showed a reduction in lung cancer incidence, new lung cancer incidence with canakumumab. We thought, well, this is rather coincidental. So we looked into it in more detail. IL-1 beta is being released by the macrophages, by the epithelial cells, we think, and that stimulates we think, the transdifferentiation of a small population of cells in the lung into a progenitor state. But on its own, it won't proliferate competently. If, however, that original cell that begins the transdifferentiation process harbors a mutation in EGFR or KRAS, off it goes and forms a tumor. So the question is then, where do these mutations come from? And the answer is they occur as a natural process of aging. So you don't have to invoke a tobacco exposure or a direct mutagen. It is just a function of aging. As you get older, you get more of these mutations. So what does that tell us about tumor initiation? If we think about the main classical model of tumor initiation, it's based on environmental carcinogens driving mutations in DNA that result in expansion of a subclone due to a driver gene that's hit with a mutation. Take tobacco, C2A mutations in the classical G12C KRAS mutation is a prime example. The alternative model is one first proposed by Isaac Berenblum in 1947 that proposes a 2-step model. 1, you need the initiator, 2, you need the promoter. The initiator is the mutation, the promoter is in this case, pollution. Now, the initiating step, obviously it's a mutation, but that doesn't have to come from an environmental carcinogen. As I said, it can be part of the natural process of aging. So I think our pollution work best conforms to that Isaac Berenblum model, recently substantiated by Alan Balmain and colleagues over the last decade and a half, who've really been pioneering work in this area and showed actually that 17 out of 20 environmental carcinogens in mice don't induce DNA mutations. So this may be a more common mechanism of tumor initiation than perhaps we once thought. And actually, it's just interesting to note that, Isaac Berenblum did these experiments in 1947, 75, 80 years ago. And to some extent, perhaps we've been, how can I put it, slightly distracted by the tobacco carcinogen DNA mutation origins of cancer, which clearly, happen, and perhaps ignored some of these earlier experiments where, which might be more applicable to the majority of environmental carcinogens. I don't know. I guess time will tell.